Myotonic dystrophy type 1 is a debilitating disease that causes muscle weakness and wasting, eyesight problems, and also heart defects, and there's currently no cure. It's a genetic condition which is linked to a gene called DMPK. This causes a toxic form of the chemical RNA, which is a genetic messenger molecule a bit similar to DNA, to build up in and compromise affected cells. They do this because the RNA molecules are sticky and they clog up other important processes inside cells. But now scientists at the University of California, San Diego have successfully used the gene editing technique called CRISPR to reverse the disease in mice, as Eva Higginbotham heard from Jean Yeo.
So in the paper, we focus on testing the idea that we can use CRISPR-Cas9 proteins to bind these toxic RNAs. We fused the CRISPR-Cas9 protein with endonucleases, meaning little enzymes that like to chew up RNA. And by recruiting the CRISPR-Cas9 proteins to these toxic RNAs, our endonucleases chewed up these toxic RNAs and freed up the proteins that were trapped in the toxic RNAs to return them to their normal functions, thereby fixing the disease. And we wanted to test this idea in a mouse model of DM1. And so to show that we can use our potential therapeutic, we encoded the therapeutic in adeno-associated viral vector, or AAV. This packaging of our therapeutic enables us to inject the virus into the mice, and the virus spreads to relevant cell types and expresses our enzyme in the cells and then destroys these long repeats in the same cells.
Were the mice then able to start functioning again as a normal mouse? Did they regain muscle strength and everything?
Yes, so the repeats were eliminated and the proteins that were trapped are restored. The mice then behaved normally, they regained muscle strength, and they, for all intents and purposes, look like a normal behaving mouse.
How do you measure muscle strength in a mouse?
By letting the mice hang on to the cage and seeing when the mouse lets go or drops off because it gets tired. Other ways in which you can measure muscle strength is to look at electrical activity in the muscle fiber, and that is also recovered in these mice. As this CRISPR-Cas9 enzyme is bacterial in origin, we were very worried that there would be immune reactivity by the mouse to this foreign protein. So we treated the mice with immune suppression for about 2 weeks while the virus was administered. And that was able, we believe, to suppress the immune reaction sufficiently so that once the immune suppression is relieved after 2 weeks, the therapeutic was able to do its job and the mice did not present with overt immune reactivity that would cause any severe side effects.
Would you have to keep injecting the mice with this therapeutic, again and again to make sure they didn't keep building up the toxic RNA?
The way we envision the therapeutic to work is that AAV, or the virus-mediated introduction of the RNA targeting CRISPR-Cas transgene, will be present in the cells for the lifetime of the cell, meaning that while the target cells are expressing the repeats, the CRISPR-Cas9 technology is in the cell eliminating the repeats. So the goal here would be to have a therapeutic that is a single dose and that this approach will enable the treatment of the disease. The next step would be to evaluate the safety and toxicity of this approach in larger animals. And I think that is very critical to find the correct doses such that we provide therapeutic benefit without negative side effects.
Jean Yeo at the University of California, San Diego. That paper was recently published in Nature Biomedical Engineering.